Hello, everyone. Welcome to today's webinar. I'm Britt Erickson, Senior Editor at CNEN, and I will be moderating today's event. This webinar is titled µSEC-MALS. Micro-UDAWN adds multi-angle light scattering to UHP-SEC for measurement of absolute molecular weight and size. It is being sponsored by Wyatt Technology. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please use the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNEN online after the live webcast. The presentation today is being sponsored by Wyatt Technology, the recognized leader in cutting-edge light scattering instrumentation and software for determining absolute molar mass, size, charge, and interactions of macromolecules and particles in solution. During the presentation, we will hear from Vincent Shia, a senior member of Wyatt's R&D team, who works on advancing state-of-the-art analytical light scattering instrumentation for characterization of proteins, polymers, and nanoparticles. Vincent holds a BS in electrical engineering from National Taiwan University and earned an MS and PhD degrees from the California Institute of Technology, where he concentrated on optical information processing. Vincent carried out postdoctoral research at Yale University in the Department of Biomedical Engineering, combining his interest in the applications of photonics with the fields of biology and chemistry. He joined Wyatt Technology in 2007. And with that introduction, I will now hand it over to Vincent to get us started.
Well, thank you, Britt, for that introduction. Good day, everyone, and welcome to the webinar. It is my pleasure to give this presentation. In this presentation, I'm going to talk about a new multi-angle light scattering detector, µDAWN. µDAWN has such a small dispersion volume that it is suitable for use after UHPLC-based size-exclusion chromatography. We call this combination µSEC-MALS. Size-exclusion chromatography, or simply SEC, separates polymers by their hydrodynamic volumes. It is an important chromatography technique for measuring molecular weight and molecular weight distribution. It is truly a workhorse in any bioprocess analytical toolkit, capable of detecting aggregates and fragments in therapeutic protein products. SEC is also routinely used for detection and characterization of particulates, process-related impurities, higher-order structures, and degradation studies. The development of SEC for the past few decades, however, has been slow or even stagnant at times until the recent introduction of UHP-SEC by Waters Corporation a few years ago, approximately 2011 for biopolymers and 2013 for organic polymers. Okay, so let's talk a bit about UHP-SEC, or ultra-high performance SEC. The most distinct feature of UHP-SEC is the usage of much smaller silica beads, about 1.7 to 2.5 microns, as the matrix of the stationary phase, compared with the 5-micron beads used in the conventional HPSEC technology. Pushing mobile phase through such columns requires much higher pressure in general. For example, if you're using a BH200 column, short column from Waters, at a typical flow rate of 0.3 to 0.4 mil per minute, that would typically generate 200 bars of back pressure. But in return, we do get greater resolution, faster results as we see here, and higher detection sensitivity with less solvent usage, and more importantly, smaller sample quantity. Similar to traditional SEC, when UHP-SEC is used with a concentration detector alone, either UV for proteins or differential refractive index detector, or DRI, for synthetic polymers, molecular weight is estimated from the sample's elution volume. We'll use these three chromatograms as our example. The general rule of thumb is that larger molecules elute earlier than smaller ones. The elution volume is then compared to the column calibration curve to estimate the peak molecular weight. The column calibration curve was generated from a panel of known molecular weight standards. Now, why is this approach not sufficient? Well, if we're using column calibration, we actually are making a few very important assumptions. For example, we assume that the sample molecules have the same conformation as the molecular weight standards, and that there is no interaction between the column material and any of the molecules. Quite often, one or more of these assumptions are not valid, and the estimated molecular weight can carry a large error. Let's take a closer look at the chromatograms of these three proteins. According to their elution order, we would think protein 1 has a slightly higher molecular weight than protein 2. And protein 3 must have a lower molecular weight than the other two proteins. So you can see that after we add the multi-angle light scattering detector to determine the absolute molecular weights of these three proteins, we found that protein 1 actually has the lowest molecular weight of the three, and protein 2 and 3 have very similar molecular weights. So it turns out the column interaction plays an important role in the elution order of these proteins. Well, since light scattering is essential to measure correct molecular weight and size for SEC peaks, I will give a very brief overview of the two light scattering techniques for online biophysical characterizations here. The first technique I'm going to talk about is static light scattering. It measures the average intensity of the light scattered by a sample at a few scattering angles as the light shines through the sample volume. Nowadays, people prefer to use a laser beam as the light source because it has very well-defined polarization and a very stable intensity. Because the scattered intensity is measured at multiple angles, this technique is also called multi-angle light scattering, or MALS. Since the scattered light intensity, as we show here, is proportional to both the mass concentration and the molecular weight of the macromolecules, we can calculate the molecular weight if a concentration detector is used together with the MALS detector. I do want to point out that when the molecular size is bigger than about 5% of the wavelength, in our system we use wavelengths of 660 nanometers. So if the molecules have a larger radius than about 10 to 15 nanometers, the MALS detection can simultaneously measure the root mean square radius, or RG, from the angular dependence of scattered intensity. Typically, when the molecules get bigger and bigger, they scatter more in the forward direction and proportionally less in the backward direction. Here, we want to emphasize that both the molecular weight and the root mean square radius are measured based on first physical principle. There is no assumption or molecular weight standards are necessary. So the next technique, the second technique, is dynamic light scattering, or DLS. Very often people also refer to this technique as QELS, quasi-elastic light scattering, but they're really the same thing. So in DLS, instead of the mean intensity, it actually measures the fluctuations of the scattered light intensity from sub-microsecond to much larger time scale. So it's the fluctuation and not the mean of the intensities that's being measured. Since the intensity fluctuations are a result of the molecular Brownian motions, the diffusion coefficient of the molecule can be determined. The hydrodynamic radius RH can be calculated from the Stokes-Einstein relationship, as we show here to the right. Okay, so that's a very brief introduction to the two major light scattering techniques that we used in our instruments. Both have actually been extensively covered and explained in our earlier webinars. These webinars are archived just in case you would like to go over them. Please feel free to review them on our website. Now, here's a flow diagram showing a comprehensive micro-SEC-MALS system with UV, MALS, DLS, and DRI detectors following the UHP-SEC separation. We have learned that in order to measure the molecular weight of each eluting species, we need to know the concentration as well. For molecules which absorb in the UV spectrum region, we can measure the concentration with known extinction coefficients. We can also use DRI as the concentration detector. The corresponding factor is the DNDC value, which does not vary too much from protein to protein. The combination of UV and DRI detectors enable additional applications and analysis, such as protein conjugate analysis, which we will explain in a later slide. But a few years ago, when we first attempted to use the standard MALS detector following a UHP-SEC system, we encountered some challenges. Well, as we know, a typical eluted peak in the UHP-SEC system has a peak volume on the order of hundreds of microliters. This is actually much larger than the system volume of a standard MALS detector. And the dispersion introduced by the MALS detector is very well tolerated. However, the volume of a UHP-SEC peak is much smaller compared with a standard MALS detector, and significant loss in the peak resolution is expected. Here we show that the 5 sigma peak width is actually around 40 microliters. The full width half maximum is on the order of 25 microliters, just for your reference. As you can see clearly in this example, the resolution from the UHP-SEC separation is seriously compromised by the excessive dispersion coming from the standard detectors, to the point that the fragment peak is completely lost in the downstream DRI detector. So the obvious first step is that we must reduce the volume of the MALS detector to be compatible with the UHP-SEC peak volume. And that is what we did. The picture here shows how much smaller the microdon flow cell is compared with the traditional MALS flow cell. Including other necessary components and tubings, you see that we were able to reduce the MALS system volume by a factor of more than 10, and this goes a long way in preserving the resolution of the UHP-SEC peaks. I have shown that in the cartoon here. So you see the peaks are much better resolved in the microdon system. In addition to the size of the flow cell, the optical configuration of the microdon is also different. Turns out we had to redesign the whole optical system to make this dramatic reduction of the system volume possible. And just like our other MALS detector, a DLS detector can be added for the measurement of hydrodynamic radius. So now we can compare the molecular weight from these two sets of detectors for an IgG sample with a fragment. Look at the blue curve here. The excessive dispersion from the standard MALS and DRI detectors gave incorrect molecular weight trend across the peaks. On the other hand, the red curve shows that with the combination of microdon and UT-REX, which is our low dispersion DRI detector, the peaks are better resolved. Proper band broadening algorithm can be applied, and the molecular weight result is much more reasonable. An important implication is that all key online MALS applications can now be made possible on the UHP-LC platform. Okay, so now let's look at a few examples. As the first example, let's look at the micro-SEC-MALS instrumentation applied to the reference material A671 released by NIST in 2016. It is a humanized IgG1 kappa monoclonal antibody. Extensive biophysical characterizations of the NIST MAB was published in an ACS symposium series, which I have shown at the bottom of the slide. And I also show here the SEC-MALS results from page 259 of the symposium. We can see that UV is used as the concentration detector. The MALS readily measures the molecular weights of the dimer and the monomer. Notice that at a flow rate of 0.4 ml per minute, it took more than 40 minutes to complete the sample run. By the way, they used a TSK gel SEC column with 7.8 millimeter ID, 30 centimeter length, particle diameter of 5 microns. Well, the same study also ran the NIST MAB in a Waters BH200 SEC column with particle diameter 1.7 microns, and the result is shown here. It is immediately clear that the UHP-SEC separation is much faster. At the same 0.4 ml per minute flow rate, the analysis is complete in less than 5 minutes. Quite impressive. In addition to the aggregates and the monomer peaks, you can see a low molecular weight fragment peak. Had the researchers had a microdon at the time, they would have been able to measure the species molecular weights online. So we thought we would complete the picture with a micro-SEC-MALS run, and we did just that. So now you can see that we can measure the molecular weights of the monomer and dimer in a UHP-SEC system. In addition to the measurement of monomer and the dimer's molecular weights, the DLS detector in the system measures the hydrodynamic radius of the monomer to be about 5.5 nanometers. This agrees very well with the results from sedimentation velocity experiments published in the same series. We explained earlier that MALS can measure the size of molecules if the molecules are large enough to give angular dependence of the scattered intensity. since DLS is able to size molecules smaller than 10 nanometers, actually down to maybe even smaller than 1 nanometer, it is a valuable and complementary addition to the MALS detection system. For larger molecules or polymers whose mean square radius can be measured by MALS at the same time, measurement of the hydrodynamic radius can give information on the shape and conformation of the molecule. Now we can turn our attention to the fragment peak. Since the mass fraction of the fragment peak is only about 0.1% of the total eluted mass, we really need to zoom in on the peak to get a better look, and it's shown here. Assuming the DNDC value of the fragment is the same as the monomer, microDAWN measures the fragment molecular weight to be about 50 kilodalton. These measurements are direct results of the sensitivity and resolution of the µSEC-MALS system. So indeed, we're able to actually fill in the blanks. We did something that wasn't possible before. One last slide about the NIST map. Since µSEC-MALS has the advantage of fast and high-resolution analysis, it can be used effectively as a tool to monitor degradation after the sample has been exposed to various stressors. Here we show that the µSEC-MALS results of the NIST map as it is being thermally stressed at 62 degrees Celsius. We chose this temperature because it is just a few degrees below the NIST map's first melting temperature measured by DSC. All the measurements were measured in triplicates, and we did keep a fresh vial of NIST map at 4C as a control every time we make the injections. We can see that as the sample is being stressed, the mass fractions of the aggregate and fragment continue to increase. Interestingly, as you can see, another fragment peak here, closer to the monomer peak, becomes more and more pronounced, and its molecular weight is measured to be close to 99 kilodalton. More details are available if you're interested in our 2017 publication in the scientific reports. It is open access, and you can read it through the link as shown here on the bottom right. Okay, so here's our second example. Since stability is directly related to efficacy and safety of a therapeutic protein, it is very often studied under stress conditions, such as by temperature stress, light, pH, and mechanical stressors. And when you do the measurements, sometimes µSEC-MALS can reveal the presence of unknown species. These unknown species UV extinction coefficients can be unavailable, so DRI in these cases should be used to determine their concentrations for the molecular weight measurements. And since we have both UV and DRI detectors online, the UV extinction coefficients can actually be measured from the recovered mass. In this particular example, that's exactly what's done here. So we were able to identify IgG and its various fragments by their molecular weights and extinction coefficients. The calculations are done by the Wyatt Astra software. Sensitivity and reproducibility are important aspects of any detector for sure. So we tested a mixture of two proteins at 5 different loadings from 0.4 to 6.4 micrograms. Each injection amount was repeated 3 times. Molecular weights from these injections are very consistent, as you can see, by the flat molecular weight across the peaks, even with the lowest loading, the 0.4 micrograms loading. The light scattering signal is strong. For a signal-to-noise ratio of 10, which is usually defined as the limit of quantification, we can even lower the injection amount further to 40 nanograms for this particular mixture. These results confirm that microdAWN is really among the most sensitive MALS detectors in the world. Here we also have a table that summarizes the measured molecular weights of these two proteins with the different injection amounts. The relative standard deviation, as you can see here, is less than 1%. These results show that the measurements by microdAWN and UT-REX are robust and highly reproducible. Our next application is the protein conjugate analysis. So one of the most powerful applications of SEC-MALS together with UV and RI detectors is its ability to characterize protein conjugates. And now you know this application can be readily transferred to the UHP-SEC world. Here we show just that, the micro-SEC-MALS results of a glycoprotein from a virus surface. The UHP-SEC resolved at least 3 different peaks, and the microdAWN, UV, and UT-REX measure the molecular weight of the conjugate. As you can see in the top line, it measures the molecular weight of the protein, the middle line, and the glycan component, the bottom line, across the peaks. These molecular weights suggest that the main peak is a monomer, and the earlier eluted peaks actually correspond to dimers, trimers, and possibly even tetramers. The last protein application is unique for UHP-SEC. As we know, SEC is very often used to analyze fractions collected from a prep scale HPLC during protein purification or manufacturing. Since it usually takes about 15 minutes or even longer for each analytical SEC run to complete, it can only be used offline. UHP-SEC can reduce that time to 4 to 5 minutes or even shorter, making it possible for at-line process monitoring. The addition of microdAWN can increase the capability by identifying the aggregates, fragments, and possible impurities. Here you can see that how the micro-SEC-MALS further characterizes each sample slice from the prep scale SEC run, and apparently the mass ratio of the aggregates decreases as you push from slice 1 to slice 3, and you can also look at the fragment mass fraction here. So now, to summarize what we have, microdAWN has overcome the long-standing and stubborn barrier to microliter-scale peak volumes, and the micro-SEC-MALS is faster, using less sample, and has higher resolution compared with conventional SEC-MALS analysis. Now it is possible in UHP-SEC to make online absolute molecular weight and size measurements directly in the mobile phase. The sample is fractionated. Well-established SEC-MALS applications, such as protein conjugate analysis, are now transferable to UHP-SEC platform. We also just talked about a potential unique application for process monitoring or at-line monitoring. I hope this webinar presentation is useful and interesting to you. For more information, please feel free to visit our website to learn more about light scattering theories and wire products. You can also download application notes, search for peer-reviewed articles, and listen to archived webinars on demand. I would like to acknowledge here the WIRE team for generating the beautiful data used in this webinar, and thank you for listening. I'm now ready for questions.
It's now time for the Q&A part of the webinar. As a reminder, to ask a question, please type your query into the Q&A box. And give me just a minute, and I'll check the queue to see if we have any questions coming in. Okay, Vincent, is a reference molecular weight standard required for calibration test samples' molecular weight?
Oh, I can answer that question. So, as we already talked about in the webinar, no molecular standard is necessary to calibrate for the molar mass results. However, our instrument, since it's a light scattering instrument, it does measure the Rayleigh ratio. That's another way of saying the signal, the scattered light signal from your sample. We do need to give a calibration constant for the light scattering instrument to go from the voltage that it gets from the light scattering to the Rayleigh ratio used for calculating the molar mass of the macromolecules. And the standard that we use is HPLC grade toluene, because its Rayleigh ratio is very well documented, very well measured, and that's the only calibration we will need for microDAWN, or for, in general, the MALS instrument.
Okay. For the NIST MAB, could you resolve the different masses of the 3 to 4 high molecular weight species that were present?
Great question. We could definitely resolve the dimer, as you can see in the presentation. We could resolve almost a trimer, but anything higher than that, because they are quite gobbled together, we couldn't really tell, have a very flat or very well resolved molecular molar mass results. And that if somehow we, in the future, the different species can be resolved better from the SEC column, then most likely we'll be able to resolve those species. So this is not a limitation of light scattering, but rather the separation of the different species. Actually, higher molecular weight species, they scatter way more light than the lower molecular weight species. So as long as we can measure the concentration of them, it's really easy to measure the molar mass of these higher order aggregates.
Okay. When there is a difference in RH versus RRMS for the molecule, it's different. Sorry if I'm not making much sense here. 9 nanometers versus 11 nanometers. What can be considered correct, or what other information about the molecule can be inferred? Did that question make sense?
Sure, yes.
Okay.
Thank you. So there are two different radii that we have mentioned in the webinar. The RMS, let's review a little bit. RMS is the root mean square radius. So basically, that's a way of measuring how the mass of the molecule is distributed. If you think of the molecule as a square, as a sphere, as a solid sphere, the distribution is uniform throughout the whole sphere. The root mean square radius would be about 78% of the way from the center of the sphere. That's because that's on average all the mass is. That's like all the mass is located as far as the sphere is concerned. The hydrodynamic radius is a little different. It is the radius of a sphere that would diffuse as quickly as the molecule. So imagine now you have let's stick with the sphere. If you have a sphere, the hydrodynamic radius would be very close to the radius of the sphere. However, the root mean square radius would be smaller than the actual radius of the sphere. So in general, the hydrodynamic radius would be slightly larger than the root mean square radius. However, if your molecule is very extended, they have mass way far from the center, if you will, of mass of the molecule, then its root mean square radius can be larger and larger. So now you probably can imagine, if you know the ratio of the hydrodynamic radius and the root mean square radius, you can infer more or less the shape of this molecule. So that's the difference between these two radii. And the question says if the root mean square radius is slightly larger than the hydrodynamic radius, what does it mean? That probably means the molecule is very highly branched, and it's got components that's distributed very far from the center of mass.
Okay, thank you for that. Could this technique be applied to synthetic polymers?
For sure, yes. So we have routinely measured polystyrene. We have, yeah. So the answer is yes. And you can also run microdon with APC, so no problem. Organic solvent is also compatible with microdon.
Okay. Is UHP-SEC applicable to nanoscale systems?
Oh, good. That's so nanoscale system. I suppose you're referring to nanoliter volume. So it comes back to the dispersion again. Microdon has a cell volume of about pi microliter. You saw that in our presentation. The total system is about 8 microliter. So anything much smaller than that, the peak will be dispersed pretty badly. So maybe that's our next goal, to develop a nanodon. I do not know who asked this question. I am very interested in knowing your application. Please feel free to contact me. Let's talk about your application, and it might be a fun project for us.
Okay, great. Does conformation affect the molecular weight determination, and why, for synthetic polymers?
The conformation does not affect the molar mass measurement. So in MALS, when we make a measurement of the molar mass, it's a global fit of two important parameters. One is the molar mass. The other is the root mean square radius. So these two components are not independent of each other. To determine, so if you have a synthetic polymer that is bigger than, say, 10 nanometers and it's got an angular dependence, the calculation of molar mass must take this angular dependence into account. So what our software does is it automatically extrapolate or fit the angular dependence so that we know at the limiting angle of 0 degrees what the Rayleigh ratio would be, and that is exactly the definition for the Rayleigh-Gann scattering, how we can calculate the molar mass. So no matter what the shape of the polymer is, as long as its root mean square radius and the Rayleigh ratio can be measured correctly at the 3 angles that microDAWN has, then there's no problem measuring the molar mass or the root mean square radius of the molecule.
Okay. Are the samples more likely to be degraded by higher shear in the UHP-SEC columns?
Interesting question. So the linear flow rate in UHP-SEC versus HP-SEC is about, it's probably 3 or 4 times faster in UHP-SEC. So it is not that much higher, and from our experience with UHP-SEC sample runs, we don't really see such an effect. But if you suspect your molecules are very fragile and can be degraded by shear, you can do the following experiment. You can run UHP-SEC at different flow rate. In our case, if you have a BH200 column, you can run anywhere between 0.05, we actually went down to 0.05 mil per minute, up to even 0.6, 0.7 mil per minute. And if there is shear degradation, you'll probably see a difference in the different components. For example, the fragment peak or the higher molecular weight peak. We don't see that. So there's that is quite satisfactory to us that there should not be shear degradation. Sorry.
Okay, great. Thank you. What is the lowest difference limit in molecular weight for conjugated samples versus unconjugated samples that can successfully characterize using DAWN to Helios and microDAWN?
Good question. So the conjugate analysis. So let's talk about the molar mass determination of MALS. MALS and mass spec are different. MALS measures the molar mass from the Rayleigh ratio, the light scattering intensity. So its accuracy or its precision can be really good within 1%. Its accuracy, depending on the system, could be about 3 to 5%. So that's a rule of thumb. In order to distinguish the glycan component and the protein component of your conjugate molecule, it is good to keep the precision about 1% in mind. So, so that combined with the fact that the molar mass range measurable by the MALS technique in microDAWN is anywhere between 200 Dalton to, depending on the shape, a few million Daltons. And in Helios, if you have 18 angles, the molecular range can be extended, the upper end mostly to gigadalton range. So if you have a conjugate that the glycan component is more than 2 or 3% of the protein component in terms of molar mass, you should be able to pull that out. But if you have just one the glucose attached to a big molecule, big protein, 150 kilodalton, you will not be able to see that. So it all comes down to the resolution, the precision.
Can you explain more about the extinction coefficient determination?
Sure. So that's when you have two different concentration detectors, and they are looking at and their response factors are actually different. For example, for UV, in this case, it's looking at the aromatic residues of the molecule, of the proteins, that is, the tyrosine, the tryptophan, and the cysteine, I think. So that's the extinction coefficient. The DRI detectors, it's not looking at these aromatic molecules alone. It's looking at all the residues because all of them have their associated refractive index increments. So now, as it turns out, for the DRI, most of the amino acids have very similar DRI response. So most proteins have very similar DNDC values, and 0.185 is very, very often used if you're not sure. And it's usually a very good guess. For example, in the NIST MAP case, we started out with 0.185, and then in the end, it's about 0.187. And I'll tell you how we determined that. So now, if you have an unknown protein and you don't know you do not know its UV extinction coefficient, a way to try and determine that is to first assume it's 0.185, and it's usually a very good guess, usually within a few percent. So assuming its DNDC value is 0.185, and by looking at the RI trace, you can integrate over the RI peaks and figure out the recovered mass concentration from that. So now you know the total recovered mass of your protein. Let's say it's 100 micrograms that's recovered from your SEC-MALS run. You can then determine what would the extinction coefficient be such that you get the same recovered mass for each of the peak, if you will. And that's how you could determine or have a very good guess, very good guess of the extinction coefficient of each of the species in there. In the case of the NIST MAB, 0.185 is a really good guess, but since we know the extinction coefficient of NIST MAB, it's published by NIST, it's 1.42, I believe. So if we use that so that would be the other way around. You can figure out the recovered mass from your UV signal, and then figure out what the DNDC value should be to reach the same recovered mass. And turns out it's not 0.185. It's very close. It's 0.187. It's about 1% off. But either way, you should be. It's really quite acceptable in the case of molar mass determination with MALS.
Okay, interesting. What column was used for the glycoprotein? Is there any column that's good for greater than 1,000 kilodalton conjugate?
So I'm not quite sure of that question. I'm sorry, I cannot answer that. So, but I'll get back to. I'll make sure to find out and get back to you. I'm sorry about that.
Okay. No worries. Let's see what else is in the queue. One moment, please. What is the measurable size range of microDAWN?
The measurable size range of microDAWN. So the hydrodynamic radius is anywhere above about 1 nanometer to 30 nanometers at a flow rate of 0.3 ml per minute. So that's hydrodynamic radius. The root mean square radius is determined by the number of angles and the actual values of the angles used in microDAWN. And since there are 3 angles in microDAWN, the measurable RG range is up to 50 nanometers. That's posted on our website as well. And the sample we use to confirm that is actually a 1.6 million Dalton polystyrene in THF and as well as in toluene. So that's the size range measurable by microdon.
Okay. How do you calculate the molecular weight of pegylated protein?
The molecular weight of the pegylated protein. Yes. So let's start from. So it's really the same principle as light scattering. In the slide, we mentioned that we measure the Rayleigh ratio, and then we measure the concentration of the protein as from the concentration detector. Since the Rayleigh ratio is proportional to the molar mass, the product of the molar mass and the mass concentration of the protein, then we have one unknown, one equation, we'll be able to figure out the molar mass. In the conjugated or pegylated protein case, now it's the same principle. So the glycan component and the protein component, they both are scattering light. It's just a ratio. Which one scatters how much we do not know. That means we have two unknowns, two unknown molar masses. And in order to resolve these two unknowns, we need two concentrations, right? And that's where the two different concentration detectors come in. The UV is sensitive to the protein component alone. So from the UV, you can easily determine the protein concentration. The RI is sensitive to both the DRI detector is sensitive to both the polymer and the protein component. So it's measuring the concentration of these two together. So now that you have two concentrations, you can deconvolve, if you will, these two concentration components, and you can pull out the respective molar masses of these two different components of your conjugate.
Okay. What is the upper limit of measuring higher molecular weight?
The upper limit of the molecular weight is mostly determined, in our cases, the number of angles. So trials and microdon, they both have 3 angles. They're both 3-angle MALS detectors. The RG that could be confidently determined in these two instruments are, as we mentioned, 50 nanometers. So depending on the shape, the molar mass is up to, depending on the shape, in the 10 million Dalton or so. If you have more angles, you'll be able to determine the root mean square radius better, or extend the upper size range measurable. So Helios, we have 18 angles, and we'll be able to measure 500 nanometer RG, and depending on the shape again, so that would be in the 2 gigadalton, 2 gigadalton range for the molar mass.
Okay. Can you remind us, how do you know the DNDC value of your sample?
Oh, for sure. So that's a good question. The DNDC value can be determined many ways. I mentioned that if you don't know and you just want a shot in the dark, 0.185 is great for proteins. We know polystyrene in toluene is about 0.11 as well. So the first way is to, from experience, you can have a very good educated guess. So that's one. Another way you can there are other ways. For example, you can determine DNDC experimentally. If you have an unknown sample and you would like to a sample of unknown DNDC and you would like to determine the DNDC of it, you can make a series of samples in different of this
unknown sample in different concentrations. And then in the mobile phase that you would measure it in, it's a very good idea to dialyze your sample against the mobile phase you're going to be using, because that's actually quite important. You then inject this series of concentrations into your DRI detectors and record the RI, or the refractive index signal, of these different concentrations, including the baseline, that is just the mobile phase alone. The refractive index, as a function of the concentration, should give you a straight line. So the slope of that line is giving you the DNDC value. We recommend you use at least 3 to 5 different concentrations to get a good confidence level. Another way you can also determine the DNDC is by running the sample through a column. So the column is very advantageous in this way because it's basically dialyzing the sample for you as it runs through. So in this case, you can also you can do it 2 ways. One is you inject a certain amount of the unknown sample and you assume 100% mass recovery, and by that you can then figure out what DNDC value would give you 100% mass recovery. That way you can measure the DNDC value. The other way, as we also mentioned earlier, is that if you know the extinction coefficient of something, or you have of the unknown sample, you can then run it through 2 concentration detectors and use 1 concentration detector to determine the response factor of the second concentration detector, in this case the DRI. The advantage of doing that is you don't have to make assumption that you have 100% mass recovery, although you do need to know the extinction coefficient of the sample that you're using. But in any case, these are several ways you can determine the DNDC value of your proteins, your polymers.
Okay. And I think we have enough time for maybe one more question.
Okay.
Can UHP-SEC with microdAWN be used for carbohydrates?
Oh, yes. Yes. So if you know the DNDC of your DNDC of the carbohydrate that you're interested in, then yeah, definitely you can certainly use it for carbohydrate. That's a great question, actually, because that's also because you probably cannot use UV to determine the concentration of your carbohydrate. So RI detector would be preferred in that case. Thank you for all the wonderful questions. We couldn't get to all of them, but I'll make sure to find out each and every one and get back to you guys. Thank you for listening.
Okay, well, that's all the time we have. Thank you again, Vincent, for your fascinating presentation. And thank you, participants, for being a great audience. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. Thank you, On24, for technology and production services. And thank you, Wyatt Technology, for the sponsorship that made this interactive webcast possible. For CNN webinars, I'm Britt Erickson. Goodbye.